Hi, my name is Charles Cuttino, and I work in the Schroeder Lab this summer, and I'm currently an undergrad at the College of Worcester, and my project is investigating how glucose regulates protein modifications to influence longevity in C. elegans. So diet plays an important regulatory role in the longevity of the nematode C. elegans, and in particular, high dietary glucose is known to decrease the life expectancy of C. elegans due to the high dietary glucose leading to oxidative stress, and then also abnormal protein modifications that are associated with the decline of aging. However, interestingly, there is a longevity phenotype associated with glucose if glucose is only exposed to C. elegans during its larval stages of development and then revoked during adulthood. And this causes the life expectancy of the C. elegans worms to increase compared to the average normal wild-type C. elegans worms that you'll see. So what my project is kind of looking at specifically is one of the possible mechanisms behind this. So we're looking at epigenetics, which is essentially a form of just gene regulation involving histone proteins and adding tags to mark them. So we are looking at acetylation and methylation. And acetylation activates genes to allow them to be transcribed and expressed, and methylation silences them and prevents them from being expressed. And we're going to be looking at that and investigating that for my project. And basically how I did this in my methods, I was able to culture C. elegans on 3 different sets of plates. One set of plate didn't have any glucose on it, so the worms just grew. I grew the entire experiment without glucose, and this was considered the control. But the other 2 I was looking at contained glucose, and we cultured C. elegans in their larval stage until adulthood. And at adulthood, we transferred them onto another non-glucose or nematode growth media glucose plate to chronically expose the worms to more glucose. And then we transferred another set of glucose developed worms onto a non-glucose media plate to ultimately induce that longevity phenotype, to kind of only expose them exclusively during larval development. And then we extracted their proteins on day 1 and day 5. To eventually Western blot them to determine if there's a difference in epigenetics between day 1 and day 5 of the worm's adulthood. And my results ultimately showed that chronic glucose supplementation leads to chronic methylation over time. So worms will demonstrate methylation in their histones throughout all of their adulthood. However, when developmentally exposed to glucose only, they start off being methylated, but they slowly decrease the methylation over time, just due to, I guess, the lack of presence of glucose is what is kind of the overall conclusion of that. And then for the acetylation, we noted that there isn't any acetylation on the chronic glucose supplementation worms of the histone 3 that we were looking at, but there is acetylation of other proteins, but no acetylation of the protein, of the histone protein. But in only developmentally exposed glucose worms, we did see some acetylation of the histone protein. And what essentially all of this means is that glucose does in fact have an influence on the epigenetics of C. elegans, and that could be associated with its life expectancy and longevity. And so future work would include looking at future F1 offspring progeny to see if these epigenetic changes are possibly hereditary. So, yeah, that's my project. Thank you for listening.